Today, we're going to go through the differences and the similarities between mitosis and meiosis, which can be a difficult concept for both intro and advanced biology students alike. So, right here, we have a graphic. On this side, we have mitosis. On this side, we have meiosis. The first thing that needs to be understood between these two things is that mitosis is when cells simply replicate. This happens all of the time. This happens when you scratch your arm and a bunch of cells fall off and your body has to rejuvenate those cells. This happens all of the time. Meiosis, on the other hand, does not happen all of the time. Meiosis only happens when you have to make sex cells. Mitosis, autosomal cells, meiosis, sex cells. So, since you're making sex cells, you don't need to do this as often because you're not having children every time you scratch your arm. So, because of this, mitosis happens much more frequently than meiosis. Now, if you go through the stages of mitosis, where we can start, it is prophase, metaphase, anaphase, telophase, interphase. Okay, so an acronym to remember that is PMATI. I know that's a strange acronym and it might not mean much to you, but you can of course create a sentence with it or any other way that you find it easier to memorize it, but I can always memorize it by PMATI. And so you'll go through the stages, which is prophase is when your cell is basically just preparing to replicate because it's exactly what it's doing. It's just creating a clone of itself. So it's just condensing the chromosomes, all of that stuff. And then you go into metaphase. And metaphase is when the chromosomes get pulled apart to separate sides of the cell. Okay, you're using microtubules to do that. And then you'll go to anaphase, where it's the actual separation. Because metaphase, they're just lining up. They're preparing to separate. Anaphase, you actually pull them apart. And then you'll go to telophase, which is where the cells physically separate, meaning the cytoplasm and everything in that cell separated. In the end, you're left with two identical daughter cells. You can see this guy right here. He's the parent. Well, you got two daughters. So the two daughters are explained as 2N. So what does 2N mean? 2N means that they have they have 46 chromosomes, the proper amount, the total amount of chromosomes. The typical autosomal cell is 2N, or 46 chromosomes. So now that you have that, you can replicate these, continue these, have skin on your arms at all times. You're always having the correct amount of cells. Well, then you have interphase. Well, what's interphase? Interphase is what the cell is in at all times if it's not replicating. So if you see here, there's G1, S, G2, and M. G1 is when the cell's just doing what it normally does. Your liver cell is livering, your heart cells, hearting, doing all the stuff that it needs to do. But then it goes into the synthesis phase. This is when it's going to replicate all the DNA, because if you're creating two cells that are identical, then they have to have the same amount of DNA, which means you need to double the DNA in the parent to be able to do that properly. And then you have G2, which again, it's just functioning as normal, growing, preparing to divide into two cells from one. Then you have mitosis, which is what we just described. Now, it can be super confusing to memorize two completely different pathways for cell replication, such as meiosis. But all you have to know is it's PMATI 2 times. So basically what you're going to do is you're going to go through all the processes 2 times. And this makes sense because you know that sex cells are only 2N. They have the half the amount of chromosomes necessary because when two sex cells combine in fertilization, it creates one cell, which makes sense, which helps you also understand that meiosis is for sex cells only. So you go through this prophase, metaphase, same thing. The only thing that is exponentially different in these two phases, the two different versions of PMATI, is that in meiosis 1, it's dealing with homologous chromosomes. In meiosis 2, it's dealing with sister chromosomes. So homologous chromosomes look like this. They're sitting next to each other, and they're lining up at the metaphase plate, just as you know from over here. So these two guys sitting right next to each other. This is from mom, this is from dad. They both code for hair, but one is from mom and one is from dad. So they're homologous. Homo means same, for the same gene, but they are made for different things. So when they get separated, mom and dad, they're separated over here. So that means during telophase, when there are two different cells after the first rotation of P Maddie, it's moms on one side, dads on the other side. And then when it goes through again, it's going to separate even further. So that's how it creates the 1 N per cell. It was super helpful in explaining the differences and the similarities between mitosis and meiosis. If you need more help with the content or need help in general, or to look at more videos, please go to www.clemson.edu/ASC, because there you can find more videos, more help, and even our drop-in tutoring hours if you want more in-depth information on this topic. Thank you so much.